Hello friends, now we are going to see about plant growth and development. Coming to the introduction, all cells of the plants develops from a zygote. So the zygote produces a number of cells which organizes into tissues and organs. The development is the sum of two processes, growth and differentiation. During the process of development, a complex body organization is formed that produces root, leaves, branches, flower, fruits and seeds, and eventually they die. This picture will tell us about when we sow a seed in the soil. So this is the soil line. From the hypocotyl radicle, the root system will develop. From the epicotyl, the shoot system will develop. So later on growth and differentiation, it will develop into a complete form of the plant. Coming to the next, growth. Growth may be defined as an irreversible permanent increasing in size, volume or mass of a cells or organs or whole organism. Growth is one of the fundamental characteristics of the living beings. It is accompanied by metabolic process, that is anabolic and catabolic process, that produce it at the expenses of energy. For example, expansion of the leaf, elongation of the stem, etc. These will be defined as the growth. Next, characteristics of the growth. The main characteristics of the growth are cellular growth, cell division, cell expansion and cell differentiation. Coming to the next, growth rates. The growth rates can be defined as increased growth per unit time. The rate of growth can be expressed mathematically as we have two types of growth rates. One is arithmetic growth, other one is geometric growth. This is the arithmetic growth and this is geometric growth. So coming to the first, arithmetic growth. After mitotic cell division, only one daughter cell continues to divide while the others differentiate and mature. For example, root elongation at the constant rate. Mathematically, it is expressed as LT is equal to L0 plus RT. LT is nothing but length of the time. L0 is length at time 0. R is equal to growth rate or elongation per unit time. So there will be a constant linear growth, a plot of length L against the t. So this is the height of the plant, this is the time. Along with when proceeding the time, the length of the plant will be elongated linearly in a constant fashion. This will be defined by arithmetic growth. Next, geometric growth. Initial growth is slow, which we call it as lag phase, followed by a rapid increase in the growth, which we call it as log exponential phase, and followed by a phase where the growth slows down, which we call it as stationary phase. For example, all cells, tissues, organs show this type of growth system. If one plots the parameter of growth against the time, it would be a type of sigmoid or S curve. The exponential growth can be expressed as W1 is equal to W0 ERT. W1 is equal to final size, weight, height, number, etc. WO is equal to initial size at the beginning of the period. R is equal to growth rate. T is equal to time of the growth. E is the base of natural logarithms. So size, weight of an organ, there will be a lag phase. We have an exponential phase and stationary phase. Coming to the next, differentiation, de-differentiation and re-differentiation. What are the three differences we are going to see? Coming to the first, cellular differentiation is the process by which a less specialized cells become a more specialized cell type. So cell derived from a meristem and cambium differentiate and mature to perform a specific functions, which we termed it as differentiation. For example, initially the cell is undifferentiated plant cell, and later this cell can be differentiated into leaf cell, transport cell or any other cells. So this we call it as differentiation. Next, in the differentiation, cell undergoes structural changes during the differentiation. Changes takes place both in the cell walls as well as in the protoplasm. Examples, cells lose their protoplasm during the formation of tertiary elements. Plants develop a strong, elastic, lignocellulosic secondary cell wall to carry water to long distances even under extreme tension. Next, de-differentiation. De-differentiation is nothing but removal of somatic tissue into the cell's cultural medium and programming in under the stressful condition, we are reprogramming with auxins plus cytosins, which are the growth hormones, and later proliferation and callus formation. Nothing but in the de-differentiation is important biological phenomenon whereby the cell regress from a specialized function to a simpler state of reminiscent of the stem cells. An undividable differentiated cell sometimes regain the power of differentiation. This process we call it as de-differentiation. De-differentiation is a common process in plants during the secondary growth and in the wound healing mechanisms. So coming to the redifferentiation, a de-differentiated cell can divide and produce a new cells. So new cells produced again lose the power of division and become a part of permanent tissue, which we call it as redifferentiation. For example, formation of tumor cells. So in this picture, this is the differentiation. From the differentiation, the stem cell like state, transient state has been developed, which will be undergoing redifferentiation or transdifferentiation, or which will be undergoing to cell death, or it will again enter into a cell cycle. Next, development. Development is a term that includes all changes that an organism goes through during its life cycle, from germination of the seeds to senescence, means old age. So initially it is meristematic cell. With the plasmatic growth, it will undergo differentiation. Through differentiation, it will finally form the mature cell. So this maturation will takes place to death, or expansion, maturation to mature cell. So from here to here, this process we call it as cell division. So this is the sequence of developmental process in the plant cell. Next, plasticity and heterotrophy. So plasticity, the ability of plant to follow a different pathways and produce different structures in response to environment and phases of life. In cotton and coriander plants, the leaves of a juvenile plant are different in shape from those of mature plant. That we call it as plasticity. So we can see this. So in the young age, the leaf shape will be different from the adult. And next, heterophile, the phenomenon of appearance of different forms of leaves on the same plant. We call it as heterophyly. Heterophyly in cotton and coriander is been common. Next, plant growth regulators. Plant growth regulators are by phytohormones, are the chemical substances that influence the growth and differentiation of plant cell, tissues and organs. Plant growth regulator function as chemical messengers for intercellular communication. They work together coordinating the growth and development of cells. They could be indole compounds like indole 3 acetic acid, adenine derivatives like N6 furfuryl aminopurine, kenetin, derivatives of carotenoids like ascorbic acid ABA, terpenes, gibberellic acid GA3, or gases like ethylene C2H4. So coming to the phytohormones, we have auxins, gibberellins, cytokinins, abscisic acid, ethylene. So these we call it as phytohormones. Classification of PGRs. So we have the growth promoters and growth inhibitors. So growth promoters are auxins, gibberellins, cytokinins. Growth inhibitors are abscisic acid, ethylene. So auxins. Auxins was discovered by Charles Darwin and Francis Darwin. F.W. Went isolated the auxins from the tip of coleoptilis of oat seedlings. Went developed Avena curvature test for bioassay of auxins. So the synthetic auxins are IAA, IBA, NAA and 2,4-D. So IAA means indole acetic acid, IBA means indole butyric acid, NAA means naphthalene acetic acid, 2,4-D means 2,4-dichlorophenoxyacetic. Next, physiological effects of auxins promote the apical dominance, initiating the rooting in the stem cuttings, promote the flowering, example in pineapples, prevent the fruit and leaf drop at early stages, promote the abscission of older mature leaves and fruits, induce parthenocarpy, for example in tomatoes, uses as herbicides, control the xylem differentiation and helps in cell division. Gibberellins. So, for example, Kurosawa, a Japanese scientist, identified the gibberellins present in the fungal pathogen called Gibberella fujikori. So, gibberellins were isolated from the infected rice seedlings when treated with sterile filtrate of fungus. So, the physiological effects of the gibberellins are elongation of intact stem, dwarf shoots, blotting in the russet plants, for example, henbane cabbage, overcome the natural dormancy of bud, tubers, seeds, etc. Seed germination, induce parthenocarpy or development of seedless fruits, promote flowering in long day plants, and vernalization. Cytokinins. Scooge and Miller discovered the cytokinins. Scooge and Miller crystallized the cytokinins, promoting the active substances named it kenetin from corn kernels and coconut milk. So, because of that, they named it as cytokinins. Cyto means cell, kinins means the name came from corn kernels. So, the physiological effects of cytokinins are induce the cell division, delay the senescence of leaf, means old age of the leaf and other organs, promote the lateral bud, increase the cell expansion in dicotyledon and in leaves, promote the chloroplast development and chlorophyll synthesis. Next, ethylene. The cousins confirm the release of volatile substances from a ripened orange that hastened the ripen of storage unripened bananas. So the volatile substances were identified as ethylene. The physiological effects of cytokinins promotes senescence and abscission, inhibits the elongation of stem and roots, induce the fruit ripening, breaks seeds and bud dormancy, initiates the germination in peanut seeds, sprouting of potato tubers, promotes the root growth and root hair formation, initiates the flower and for synchronizing fruit set in pineapples. Next, abscisic acid. So, in 1963, Frederick T. Adicott and his co-workers identified abscisic acid. ABA was isolated for several abscissing in accelerating substances from the cotton plants. ABA is also called a stress hormone. Coming to the physiological effects of the cytokine, abscisic acid acts as a general plant growth inhibitor, induce seed and bud dormancy, increase the seed germination, stimulates the closure of stomata, plays an important role in the seed development, maturation, and dormancy. Coming to the next, photoperiodism. Photoperiodism is a response of plant to a relative length of light and dark periods. Some plants require periodic exposure to light to induce the flowering. Duration of dark period is equally important for flowering. Long day plants, plants that require exposure to light for a period exceeding a critical duration to induce the flowering. For example, pea, barley, wheat, etc. Short day plants, plants that require exposure to light for a period less than this critical period to induce flowering. For example, rice, jowar, cotton, means it needs short time for flowering. Day neutral plants, plants where there is no correlation between the exposure to light duration and induction of flowering. For example, cucumber, rose, and tomato. Vernalization. So, vernalization is a phenomenon of induction of flowering in plants by exposure to low temperature. It prevents precocious reproductive development late in the growing season and enables the plant to have a sufficient time to reach the maturity. Examples, biennial plants. They are the monocarpic plants that flower and then die in the second season. For example, sugar beet, cabbage, carrot, etc. Means there is a phenomenon, once they ripen and next they will die. So this is about the plant growth and development. Thank you very much. Please do like, share, comment, and subscribe to my channel. Thank you.